That's my opening 15 minutes and 23 minutes. So with that, I want to say welcome to Dr. Dierk Niessing. Come on up. So this is Dierk's second year presenting to us, and he is the head of two different labs, one in Munich and one in Ulm, which I believe, on a personal note, has one of the most beautiful medieval churches ever built. It's the tallest steeple, I believe. Yeah, tallest tower. So anyway, that's Dierk has been studying PURA and the PURA alpha for most of your career, right? 15 years. So yeah, and he runs two different labs with a number of different PhD and master's students who also support that. So Dierk is a very good friend of ours and a good friend of mine. And so I want to say welcome and thank you for coming.
Okay, yeah, also welcome from my side, and thank you very much for the invitation, and also for the kind words. I'm flattered, but I think it is a mutual thing, and all the researchers really like to come here because it's a special thing for us to meet you and to talk to you, and I think it's not a usual conference we go to. It's different, and I think everybody recognizes that and appreciates also the hospitality and friendliness of all of you. All right, so what I will be trying to do is give you an overview of what research looks like, we are trying to do, and I think you will hear a lot of hopefully interesting and understandable talks. That's the main goal, I think. We could throw as much details on you as we want, but we would completely lose you, and the goal is not to do that, right? So you have the right to ask questions if you don't understand it. There are no stupid questions, and if you don't feel comfortable to ask in front of the audience, just approach us. We're very happy to talk to you. As I said last time, we like to be talked to, okay? Good. Who was in Cambridge last year, Kingston? Okay. For you guys, I have a little quiz. So the question is, you heard already, PURA alpha and PURA. This is confusing, I guess, and we kind of tried to resolve that last time, because the question is, what is what, right? We have a gene, and that gene, there's a gene, okay, I have to describe it then. And the gene has a name, and then there's a protein product. So now the gene encodes as a building plan for a molecular machine. So it looks like that, right? The gene is like a little book where there's a description of a machine, a building plan, and this is being translated into the protein. So now we have two words. So there's PURA alpha and there's PURA. Which one is which? Any guesses? Yes? Okay, and how do you remember that?
I just do.
That's right. There's a little hint, you know, it's true. We have the logo, right? Just a reminder. So you see around the word PURA syndrome, there is the DNA double helix, and PURA is the gene. So if you're mixing it up, just look at the logo and you're finding it, right? Okay, so this was kind of to warm up, because there will be a lot of science thrown at you, and not good to mix it up from the onset, right? So Dom has shown you already that we have a number of researchers who are involved in really doing the research here, and this is an alphabetic list of clinical and non-clinical researchers, and the ones with the green dots are the ones who showed up here, you know, so it's a significant portion. Before you're too happy, I should tell you that was cheating, because, you know, some of the names are duplicated. Look at the biobank. Good. So the question is, how does it all fit together, and what are we doing, and, you know, how do you fit into this? Because of course we're all self-centered, you know, scientists are thinking about their stuff, and all of a sudden their patients and families, and of course that's not like it should be. So that's a little scheme we're trying to put it all together. Of course, PURA families are always happy, and they come first. So you are self-organized. It's fantastic. It's amazing how much you achieved in these 5 years, I have to say. So you interact a lot, that's clear. And of course, you also interact with your medical doctors, your local doctors you're seeing, and so on. And hopefully, some of them interact as well with each other, right? Exchange ideas and so on. At least that will be your hope, I guess, right? And then you have clinical researchers, people who are clinicians seeing patients, but are specialized on certain areas, epilepsy, endocrinology, and so on, and who deal with the symptoms of the PURA syndrome and try to understand it. And then we have, they of course interact, hopefully with your MD, and hopefully more often with you and your kids. And then there are the rest of the researchers, the non-clinical researchers, right? And we interact a lot, but it looks like in that scheme that we're just self-contained and don't do anything with you guys. But that's not true, I'm standing here, so there are some arrows missing, right? So we are here to interact with you. That's one of the main reasons we're here. We're also here to interact with each other and to interact with the clinicians. So you see, it's a really complex network. In the end, it comes down to you talk to people, you like people, you find people interesting, and that this just happens. You know, this is how the way it works. So the foundation will be mostly you guys. The GRN, the Global Research Network, would cover probably most of the clinical researchers and non-clinical researchers, perhaps one or the other MD, but that's rather not so likely. And not all of the researchers are included in the Global Research Network. If there's some loose interactions, they're not part of like this defined network. Okay, but why do we do all this? I think we all agree that the main question, the central question is how can we help PURA patients and their families? You know, how can we make life better? How can we improve the status quo? That's the reason for the foundation and for also the research, of course, we're doing. And there are a number of questions we have to answer or aspects we have to address. First of all, of course, one should help each other, exchange experiences, educate each other. That's what we're trying today. With some kind of background, what we're doing, what the approaches are, perhaps also what the hopes are, what the limitations are, and this kind of things. For that, we need the foundation conference today, and of course, you have the use of social media, as I heard. Another aspect would be that, of course, the PURA specialists interact and investigate the disorder, so we try to find out what's going on there. Again, that would be foundation, patients registry. That's something Rebecca will be talking about later, and the Global Research Network. But the big question is, why should we actually bother to do research on that? You know, does it help anything? Does it change anything? I would try to answer that, because the answer is not simple. It's, well, an educated guess, it will, but we don't really know. We can't promise that for sure. We're doing our best. That's probably the most honest answer we can give. But the reason behind all the efforts are really, I think it's nicely summarized by Brian Drucker's statement, if the cause has been discovered, there's hope for a cure, right? And it's not more than hope right now, but that's what we're aiming at, of course. Yeah. And actually, Brian Drucker is one of the inventors of one of the most important cancer drugs that has been on the market since a while. Yeah. It was a real breakthrough. So, we're going into more specific questions. I promise there won't be only text slides coming, as more to come. But the questions that we are driven by is, for instance, what are the cellular functions of PURA alpha? And you remember, it's the protein, right? The molecular machine, right? So what is the protein actually doing in the cell? Because if we understand what it's doing, we perhaps also understand what is going wrong if it's not doing its job anymore, right? And for that, we established model organisms and human cell culture. We'll talk about that. And then what are the differences in development? Obviously, the kids, when they're born, they look pretty much normal, some differences, and then things are changing and things are happening we don't fully understand. And of course, getting a better idea of what the things are that are going differently is important to get an approach again to perhaps counter that. And then a special feature I would like to bring up quite early already is the metabolic abnormalities. So metabolism seems to be affected. I guess most of you know that. You're the experts on that. with your kids. And seizures might be connected to that. We don't know that for sure, but that's a guess we have. And the thing is that we want to do a systematic approach and understand what are the differences, and perhaps there's a way of countering at least the seizures. That would be something we will be talking about as well. All right, so to give you some introduction, the first talk, I'll go through a number of talks you will be hearing, just to give you an overview. First talk will be given by Helen, and she's a genetic counselor here in Boston, and she'll be talking about the genetics, what are the genetics of PURA syndrome. And you see the green dot, which always means the person's present, right? So then, another thing I want to point out, although the people aren't there, this is a consortium from Denmark and also from the Netherlands. Clinicians who really spend time setting up a study, a systematic study on EEG. I'll tell you in a second what that is. There was a recruitment until the spring, and I think about 60 patients had been recruited, so that's a real big success, I would say. It's a huge number, actually. And what they look at is they look at electroencephalograms, which are actually the brain waves or the electrics of the brain. And people have known since a long time that abnormalities, also seizures and so on, can be visualized by that, by really recording these brain activities. And they want to have a systematic look if there's a common pattern and if one can distinguish things and perhaps identify features. So the idea is really to learn how PURA seizures start, how they spread, and how they stop. Right. And perhaps from that we learn something about why that is happening. Then model organisms, that's a big thing for us because we can't and we don't want to work on humans. We don't want to do experiments with humans. So we need a proxy for that. We have to have a living organism where we can kind of model or recapitulate what's going wrong, and then really look, for instance, at mice and so on, and try to understand what the differences are. And there are different model organisms, and we use a number of them. We'll show you some slides on that. I'll try to give you some idea why we need different ones, because you could also say, well, we just need mice. And that was actually the first organism we started with Jennifer. But then there are questions you can't address with that. And one thing with model organisms is, they have certain distance from us in terms of evolution. Some are, lower eukaryotic species. Some are quite close. And you can see here, I'd like to have the pointer somewhere, but I don't get it. Oh, there it is. Oh, great, I don't see it. So, should stay there. So, well, this is us. And you can see the mice are the closest, and the number 90 means that we have separated about 90 million years ago. Or in terminology of my son, that is after the advent of the dinosaurs, right? So, and then you can see that frogs are more distant, and the furthest away is the zebrafish. The ones we are using are actually boxed in, I think it's red, right? Yeah, in red. So zebrafish is the farthest away, and the question is, why do we deal with that at all, if they're so far away? We'll try to give an answer. So let's start with the mouse model. Jennifer is working on that. You see her there, and Brittany, her PhD student. I only have a picture of her with a big bagel donut. And the donut is so big that you don't see her face really. So perhaps Brittany, can you stand up and say hello? Good. Good. So mice are, evolutionarily relatively close to us humans. That's a big advantage. Many diseases can be modeled, so we can look at the mice and understand things about humans. And there are many genetic tools. That means we can really do a lot of things with that. Disadvantages are, it's really slow. They breed for a long time before the next generation comes up. It's expensive. And it's not ideal for very early developmental studies because it's in the uterus, right? So we can't really look at that. We can, but it's complex. The next model system I just want to touch, and you will hear more about that, is a tropical clawed frog. That's something we added like last year, and it's a group of Matt Geil who's doing that in the UK together with Annie, his PhD student. And the advantage of that frog is that it's really nice to study early development. You know, it's developing outside of the body, so you can look at that, and it has fast and good genetics for modification. Not so good as that is, of course, later on, it's very difficult microscopically to study that. So very early development, good with a frog, not so good with a mouse. You see already the difference, right? We have another one. Oh no, and Jennifer will give the talk because none of the two has a green dot, meaning they're not here. And Jennifer was so kind to take over and give you an overview on that. The next one, zebrafish. It's a nice little fish. You find it all over the world. And the group of Bettina Schmidt, she joined us also last year. She's in Munich, and she's working actually at a center of neurodegenerative diseases, a big center who works on ALS, FTLD, and other diseases. And she became interested in, and we could recruit her, let's say, to the, to the GRN. Advantage here is it's also great to study early development, but a unique thing is you can use this fish to screen for drugs against seizures. This has been done for other types of diseases, and what you can do is really you can have the little embryos and you can modify them in a way that they have seizures, and then you add chemical drugs, different ones, or FDA-approved drugs that have not been associated with seizures, epilepsy, and so on, but might have an effect. And you know, they would just swim in the water where the drug is in and just absorb it, and we'll see if there's a difference. We don't know if it works, but that's the model we're setting up, and we have the hopes that that could give a direct suggestion for a completely different medication. That's the hope. And in a few cases, this has really worked out nicely in other diseases. Disadvantages, it's quite distant from humans, as you saw, more distant than mice and frogs. What else? Yes, she's not there, so Marcel, PhD student from my lab, he's here. He will present, give an overview of that for you. Then, the last model system I want to mention, Lena with a glass of wine. She's also been seen without wine once in a while. And Micha Drucker, so this is really a close collaboration within Munich between Micha Drucker, who is a stem cell specialist, and our lab, who has not been specializing on that before, and we had a close collaboration to setting up induced pluripotent stem cells. You'll see in the afternoon session that it's important that we say here it's a somatic stem cell line. We'll make a distinction between germline and somatic. There are ethical issues why we should not work with a germline stem cells. So advantage is these are really human cells, right? And we can actually take patient cells and look at that and see how they develop, what the differences are. So it's very close. Disadvantage, of course, it's only cells. It's not an organism, right? It's not a human being, right? But the good thing is you can do a lot of things. There's a lot of genetic tools available, and you can use the patient cells directly. As I said, it's only cells, not an organism, so you cannot study differences in development of an organism. That's not possible there. And it's expensive, and it's really slow, as we experienced, right? Much slower than we hoped for, but we keep pushing. So these are the model systems, and I just want to add on top of that that we have a number of specialized techniques, and just mention a few. One of them is the systematic analysis of metabolites, and Dierk Adamski, who is in the audience, he will give a talk about that. And the idea there is really that you look at all the metabolites in a cell or an organism and compare normal healthy organism with a PURA mutated organism. And currently that is happening together with Jennifer Gordon. And the idea is really that one wants to observe abnormalities in the metabolism with mice, with PURA mutations, for instance. We want to do the same with the human stem cells. And then the hope is to identify strategies for treatment. I'll not go into detail, but this is a very different approach than the zebrafish, right? So we're not pushing in one direction only. Something my lab has been doing quite a bit is doing structural biology. So on the left, the thing that is kind of rocking, that's the structure of PURA alpha protein. And from that, how it looks like at atomic resolution. This is really, really, really small. From that we can infer get some ideas about what a mutation does to the folding and the building up of this protein. And another thing we are doing is actually looking at the interaction partners, or as I said here, the cellular friends of PURA alpha. So, aims are how do mutations affect the folding of the protein? What does it do to the machine? How does it break the machine? Why do these mutations disrupt the function of PURA alpha? And what are the functions that are disrupted? And from that, we hope to get a deeper understanding and then a better approach of what we can do. Another part. You will hear all that again. Learning is about repeating things, right? So don't worry if you get lost. It's a transcriptome analysis. We have the PURA mice, so there are mice who have mutations in PURA alpha, and they have lots of similarities to PURA patients. And that is, of course, a good thing, because we can look at these mice and analyze them. And Jen is doing that, and she will tell you more about that later. The thing I just want to point out here is that, as I showed you before, DNA, so that's where the genomic the information, the building plan is written down, is being translated into RNA. That's kind of a messenger to build the protein, to build the molecular machine. And you can analyze the RNA quite easily and nicely, much easier than the protein in terms of amounts. And then you can compare cells from healthy mice with cells from PURA mice, and see if the same RNAs, the same building plans, are being copied for production. And there you can see differences and also get an idea what is different, what is happening in the cells. This is a collaboration between Jennifer and Andrew, who's also in the audience, and his boss, Diana Burrell from the UK. And just to give you an idea what that is, it's really complicated. You get tons of data, gets tons of charts and things that are not easy to grasp. So it's a lot of bioinformatics that has to be done before you can really make sense out of that. Good. With that, I'm almost done. I just wanted to give you one example how things can fit together, right? I was talking about the epilepsy, which is a major issue for all the families. We learn that every time we come here. And it looks like everybody is doing his or her research, in the lab, and we're pipetting and doing our things and hearing music and not thinking about the other things. But in the end, it's connected. It's really connected. I was telling you, for instance, that the metabolomics is done with mouse samples, right? So that would be the error between mouse and metabolomics, but human stem cells we will also use for that. And I told you that we have transcriptomics, so measuring the RNAs. Andrew will be talking about. Again, it's the same for mouse, for humans, but then also the zebrafish. We'll also do that. So we have 3 organisms to compare, and if there are similarities, we can be quite sure that this is an essential part, right, that we're looking at. And then in the left upper corner, you see epilepsy cohorts. It has not been connected yet, right? But as soon as we understand some of the features that might be related to neuronal function, seizures, firing of neurons, and so on, we of course make lots of connections and talk to the clinicians and try to find out if we can put all this together into a greater concept and see if we understand what's going on in the patients. Okay, and as you can think and imagine that this is a lot of information and data and it's easily confusing. So we need a structure to put all this together, right? So we don't get lost in the amount of data. And the idea is really that we have a centralized repository for that. This is found also, first of all, in the booklet, but also on the website. That's this nice scheme Mel has done a while ago, where we have the research up there. I don't see the arrow. I think it's lost again, but I think you can see the upper panel. That's all the research I was presenting to you. And we collect all the data and put them into the patient's registry. And we build up a biobank. What that means, Rebecca will be talking about, but it's a structure we can all use for the research and to understand what the bigger picture is, right? And there has been there's still one clinical fellow of the PURA Foundation, which is Rebecca. She will be talking about that, sitting over there, has a green dot, shows she's real. And the hope is that with that, we, can make informed guesses about therapies and at least treating some of the symptoms and perhaps even more. All right, and with this, I'm done. Thank you for your attention, and perhaps there's some questions.